Okay, sorry for the delay, and let's get started now. Let's first get rid of everything here. So, yep, good. Now let's just continue where we stopped last time. But as told you, it would be a good idea if now you try it on your own computers. So, of course, this is a big data set, and you will notice part of the problem. I came back from a conference yesterday late evening, so somehow this is only half prepared. Sorry about that, but I think we'll manage, and if not, you have to think for me. And we'll see. So what I now want to do is that I quickly upload this data somewhere so that you can try the same that I've tried.
I have the data.
Okay, in that case, I was thinking ahead further than I would have thought myself. So you all have this data already? This is brilliant. I've only remembered up. Okay, so have you tried out? And it even worked. Even better, yes. Okay, then let's quickly start a new session, restart our, quickly advance to the place where we stopped last time. And today, what I thought we'll do is we talk a bit about how this clustering works, maybe get a bit into the math behind it. And then, depending how far we get, we might do a bit of math about Poisson and negative binomial distribution, but probably we'll rather do this next time. And today sticks to the biology of trying to understand how these how to actually get something out of this data before we get into the mathematical underpinnings of it. So this is what we did last time. We loaded here all this data, and it seems that you already have all this and loaded it. And my computer still needs to load it. By the way, if for any of you, your computer should be too old or too small, you can also work on the server that I have here. And I can quickly give you an account. It should take, I think last time it took something like half a minute, so should probably take that as well. So you remember we just did a bit of data wrangling here, fixing the data set. So at the end we have one big count matrix with all the cells of all these different runs in one go. What we haven't talked yet about is these are data from several mice, which we all put together. And in order to be able to distinguish these mice, have I explained to you about hashtags? I think I mentioned them briefly. So they added to the cells from each mouse, they added some antibody, which supposedly binds against some lipids of a cell membrane, so that it should stick to any cell, because this is hopefully a lipid which exists in every cell. And this antibody has conjugated to it not a fluorophore as usual, but an RNA oligo with a poly-A tail. So that afterwards, when you sequence the RNA, it just looks to the sequencing chemistry or to the 10x chemistry like a normal messenger RNA, because it has a poly-A tail. So among all our reads from the genes, we can find a few reads from these barcodes. And the trick is that for each mouse, we used a barcode. We used a different barcode, but the same, conjugated to the same antibody, so that we can later check these barcodes and compare them. That's the thing that I have put here in these extra stuff. So with read 10x data, as you can see here, it tells that there is more than one matrix per type. And if we look at what it means by this, you can see that this counts zero thing here has for each always one matrix called gene expression and one matrix called multiplexing capture. If I look at this, counts not dollar $R16 gene expression. So you see this is one of these sparse matrices with 32,000 features and 11,000 cells. And if I look at the other one, the multiplexing capture, then you see this is again 11,000 cells, but only 12 features. And now let's have a look at this thing here. And you can see here, these are the cell barcodes, and there's 11,000 columns, and these are our 5 rows, which are the 5 different barcodes. And when my colleagues ran the Cell Arranger pipeline from 10x, you give this Cell Arranger pipeline a text file which tells you which of the barcodes you have used for which sample, and so they've indicated that they had 5 samples, which they given, and they've given these 5 samples 5 different names, and depending on, and so whenever so each of these 5 different random oligo sequences is now associated with a name, and this here tells you how many reads from each count it has seen. So obviously this cell, for example, is from the middle sample, and we can try to decode Santiago's sample names. I think this stands for interferon response element marker, something which the cell makes the cell light up in case it receives interferon signal. That's why I put everything in here. This was the incubation time, 48 hours. GN, I've forgotten. Yes, I remember. That means gate negative. That was, as I said, they put in this, they had a mouse line with this, with this GFP reporter, which reports interferon signaling received. So it's a GFP behind this interferon response element, which is a promoter which becomes activated by interferon. And so they FACS sorted the cells from each mouse in GFP negative and positive. And so this is why we have this R16 and R18. R16 are the negative ones, so those which weren't showing interferon signaling, and GP, so those in 18. Quickly show you, that was the other 10x directory, where you see the positive one. So these samples are paired, and there's always one sample split by FACS into 2 compartments, and one thing we can do then later is try to figure out how actually the cells which have received interferon and the cells which haven't received interferon, how they differ in transcriptome. And then we can think about why they differ, because after all they work together, so they might actually have received or not received interferon because they were in different regions with different ambient signaling, or we might have had there might have been a difference in the cell's own ability to receive interferon, so what receptors it has. But this is sort of a biology that we can try to look later by comparing each negative partial sample with a corresponding sample half, which is positive. The next thing in this code is here SVZ stands for the subventricular zone, and STR for the striatum. So I still get always confused about brain anatomy, but you know you have these lateral ventricles, and next to these ventricles is a structure called the striatum, and the border between the ventricle and the striatum we call the subventricular zone because it's below the ventricle. And the interesting thing about the SVZ is that this is where these neural stem cells reside, which still exists in the adult brain, and which is the thing that we are all interested in. What else do we have here? The age of a mouse. These mice are all 2 months old. They have also older mice, but they are not here in our samples. Whether the mice have been happy so far or whether they've been given a stroke. ISK stands for ischemia, that means an induced stroke. And this here is the number of a mouse. So you see that the first 2 samples are mouse 1, then comes 2 samples from mouse 2, and then the 3rd sample. And this here, this must be the actual oligo that was used. Hashtag oligo 1, 2, 3, 4, 5, and this here must be a typo that it says 1 or 2. So as a little warm-up example, we can look here at our data and realize, for example, in this cell, this first cell probably comes from sample 3 because it has much more oligos from sample 3 than from sample 1, 2, 4, than from of a hashtag oligos 1, 2, 4, 5. But you see, it's not always that clear. Sometimes we have a mixture. What I think what went wrong here is that the hashtag that they put too much of these hashtag oligos, so not all of them actually stuck to cells, and some was ambiently floating around when they pooled all the 5 samples, and then of course these hashtags oligos which were floating around ended up binding to a cell from a wrong pool. As far as I've been told by my wet lab colleagues, this is a common problem, and it's always tricky to get the right amount of oligos, and we are getting better at it, and this is the first time at least it worked at least halfway, but it's still not perfect. So our warm-up thing is, let's focus on the R16 and this multiplexer capture, and what we now first want to try is we want to figure out how well this worked. So for each cell we want to know what is the, how clear is the signal which tells us which sample this is from? And we can just try, each of you can try to come up with a good plot in order to see how well this worked. So any suggestions, what would you plot, for example, or what kind of calculation would you do here? So here we want to do two things. First, we want to make a call for each cell, which sample it is probably from. So for this one, we would say this is probably sample 3 because it has much more oligos from marker oligos for sample 3 than for the other two. But here we can't really say because this it has most for sample 5, but it's the gap is a bit too small. Now I want to come up with a plot where with a manner to first assign to each cell one sample, probably the sample where it has most oligos from, and second get some kind of feeling for how certain this, this call is. Yeah. Mm-hmm. Okay, let's do that. For each cell, we calculate what fraction goes to the largest thing. Now this is so just so that you all not only listen to me, you can try this yourself, come up with a way where for each column you figure out for which sample is the maximum, and then let's divide the number for this maximum by the sum for all, by the sum over all of them. Let's maybe start off by first making what we say, first making the call, so assigning each cell to one of the 5 numbers, and then getting, in the second step, getting the quality. So asking how sure is this call by calculating what Philip just suggested. Let me see what you all do now. As usual. So, just to see, get a feeling of how nimble all of you are with programming so far. But I guess everybody still needs a bit. Does everybody have an idea how to approach it? or maybe to get to help those who don't have a good idea yet a bit, I pick one random cell here. That's usually a good way if you want to figure something out, to pick a random cell. Let's say cell number 10, so column number 10. And here now I get the first 10. Oh, I've never really realized that I only looked at the first 5. So here we see now what we have in these first 10. And here it's quite clear. This is hashtag 12. And the first question is, how do I get the maximum of a vector in R? Max, yes. So, because of course this is, so with this I get that the maximum is 13,112. Next I want to be able to say that the maximum is in, look at cell 10. What's the maximum? So next thing is, where is the maximum? So which index? What's the function to do that? Who knows? There is which max for that, where you just take this, and if I do that, then it tells me it's number 12. So if now for every cell, and the next thing I might want to know is what is the sum of hashtag oligo counts. How do I get that? You can easily guess. You have a vector and you want to add up all the numbers in the vector. Yeah, simply sum. So this is how we get the sum of this. And now we have a sum. And the last question we can, what's the fraction of HTOs that fall onto the most abundant HTO? So now this should be obvious. How do I calculate this? So without R, what would you divide by what to get this fraction? Yeah. Okay, exactly. So let's take the maximum here, and let's take the sum. So, and then we see this is 89%, so this is pretty clear. The next question I now would like to look is, for all cells, get the maximum. And so we want to do this thing here, and I want to run it not only for cell 10, but for all the cells. Now, what's the R way to run something like that? Yeah. Exactly, let's do apply. So, apply has this, takes first a matrix here. Then it takes a margin, so to tell should we run this over each row or each column, and we want it over the columns. And this means take each, so 1 is row, 2 is column, or if you have a 3-dimensional array, you could do 1, 2, 3. And you could now take, and what apply now does, it says take each column, because it's a 2, of this matrix, and apply to it this function. If I do this, then, well, I haven't given it the matrix because I added this. Then, as you can see, it gives us for each cell the maximum. So I call this now the max count. And I put the whole thing in a list or in a data frame, or let's say a table, so that we get a matrix out of the whole thing. Now I have here my matrix which tells me for each cell the maximum count. What I, of course, also would like to know which cell that is. So I add here a row names, no, column names, count to get these here. And as you can see, now I'm starting to make a table. It has here the cell name, and here it has the counts of the highest oligo. Maybe I call it highest count. The next thing I now want to know is the highest sample. So in which sample has this count come from? So what would I do? It should now be easy because we just have to modify the previous line. So what do I modify? I'm asking you guys just to make sure that you're more awake than I am. Let's try what happens if I do this. 2 to 1. Then I get, it's now for each sample, how many counts I get from this sample. But that wasn't what we wanted, because what I wanted is I want to basically now do here, this part here. You remember we first got the maximum count, and then we ask where is this maximum found, for which sample. And we did this by replacing max with which max, right? Now if I do this, it tells us for each sample, but I should keep here the 2. Now it tells us for each cell in which of the 12 samples the maximum has been found. So, and if I do this now I can see for each cell which sample it is from. Ideally, I wouldn't like to have the number of the sample, but I would replace this by its corresponding sample name. So I have my list of sample names, row names. Here, you remember this thing has in the column names. So this is a matrix, and the columns are named with the cell numbers, with the cell names, and the row names are named by this here. And if I have such a matrix with the sample names, I can now do something like 3,2,1,8,5. And now it gives me the name of the third, second, first, eighth, and fifth sample. Yeah? Which means highest sample index. If this is the highest sample index, then I can now add a column where I have highest sample name, where I simply do as I've just done it here, take the row names, and here I say take this column that we've just done here with the highest sample index. Sure. And now I have for each cell here the highest count, the index of the sample the cell seems to come from, and the actual name of that sample. So far, everybody with me? Good. Next thing we want to do, we want to get a score of how sure we are. So what we did for that is we also calculated the highest, total count for each cell. HTO count. So this is how many HTOs fell and were collected for this cell in total. So what do we do here? So remember that was what we've done here. Everybody is typing, but yep. So what do I put here? Who's still awake? Exactly, the same as before, but with sum. So, and now as you can see, I now have built a table which tells us highest count, sample index, total count, and highest sample name. And what we now also wanted to have is this fraction, is a fraction of highest, let's call this. And so this is what was before 99%. So what do we put here? Yeah. Exactly. I divide the max count. Where do I have it? The highest count here divided by the max count. The max count I've called total HDO count. So, and now I have a table here with this fraction of highest. And what I can now do to get a feeling about how good this is, as you can see, this is very mixed here. For example, here we have this very good one with 13,000 counts, and it has a fraction of 98%. And here in this first one, we have only 19%, which looks really bad. Let's have a look if this can really be as bad as it sounds. But as you see, it actually is, because all these counts are more or less equal. So what has happened here, we can already see here it's only 42 counts, so none of the antibodies actually stuck on this cell for whatever reason, and all that we sampled was just oligos floating around. While here the highest count is 13,000, so a lot of them stuck. And to me, it looks like there's an average, there's something like 100 of these HDOs floating around per cell. And in terms of ambient oligos, which don't stick to anything and then just randomly get sequenced. And once we see a few more than 1,000, then we actually can do something. And to have a look at this now, what I will now do is I make a plot where I say, I want to have the x-axis. On the x-axis, I want to have the total count. And on the y-axis, I put this fraction. And for the color, I use which sample it actually was. Then, because I already know that this will be very many cells, I say the size of the cell should be not too large. And now here we get my plot. And if you look at this, it's a bit hard to see because everything sticks here to the left. And here to the right, I see a bit little. So the first thing I do is I make my x-axis logarithmic. So plus scale x log 10. So, and now we can have a bit a look at this plot here. And as you can see, it seems that, for this to really reliably say, yes, this cell definitely came from sample 3 or from sample 5, we want to have at least more than 60 or 70% come from this thing. So somehow only those cells up where we can say have been reliably identified, while those cells down here, we can't really say where this comes from. There's now a lot of structure which we can try to figure out. For example, I made this plot before, but without color. Now with the color, I already notice that at the bottom half, it seems to be that we mainly get here these more purple things, and at the top half, we get different colors. So maybe they used more antibodies here than here. So we already see that the samples behave differently. For some samples, like the more greenish bluish ones, the thing hadn't worked well. For other samples, it hasn't worked so well. what I will now do here is I go back to my wet lab colleague and ask him, are you sure you put into each of the 12 wells the same amount of oligos? Because it looks that you got way more of these than these. And that might help them to figure out what they have to do better the next time. So this is a typical kind of quality control plot, as you will never see it in a paper, but as it might help you a lot while developing an experiment, optimizing a protocol, and so on. So we can look a lot and try to figure out what's going on here. For example, I see here these two structures, this one and the next one with a bit more, and I'm a bit confused about why there is this double hump. I'm also confused about this color thing, so there's a lot of stuff to think about. But the part which I now want to drive home here is, as you can see, we haven't used any ready-made package, but we just used our own R skills in order to get to this diagnostic plot, which can tell us a lot about how well this hashtagging, this labeling of the different sample cells has worked. And that's why it's so important to have skills like this, even if you work in the lab, because otherwise it's hard to optimize your work. So let's look a bit through this code that we've just written here, just to get just to give me a feeling of how familiar you all are with this. So the first thing I've used here, who of you had known this apply command before? Okay, that's a lot. Most people don't use apply, including this logic of that you can do it here with that you can give here an axis and sum over the axis and so on. Is everybody familiar with this tibble and mutate and ggplot? Who is? Okay.
But what do you actually think? Instead of using the apply, you could directly just make a tibble and then do group bys and whatever.
Right. Let's leave this as an exercise. I mean, there's a tendency here to make a mixture of tidyverse and old style R. Apply is old style R. And as Philip just said, what you could as well do is, for example, I use this thing. I pivot it into longer and do this. The catch with pivoting is once we go to our really big data, once we go to a big count matrix, we quickly run into the thing that this gets slow because the table gets really long. The apply itself is also slow, actually. For example, instead of doing this apply max here, what I can also do, so to get this maximum, I have different ways. One way is I take this. The other way is I take, one thing is to use apply, another thing is to use this row max function, which is in the which is, I think, in the gene filter package. Where is row max? Here in the sparse matrix stats. Package, row max, and I can use this. So, it isn't. Matrix generics, okay. Library sparse matrix. Library. So, I don't even know where it is. So if you have a very big matrix, the apply function quickly gets slow because it runs a loop, and it runs the loop within R, and R is terribly slow with loops. And this is why there's all these row max, row means, column variance, and so on functions which do this quickly. And that's a very annoying feature of R, because ideally you would say this is the logical way of doing it. In a modern programming language, you would just use the apply, and the language would say, okay, he wants to run this row by, so let's figure it out, and comes out with a quick way of doing that. In R, because this is by now a 40-year-old language. We always have to use, there's always for the same job, there's different ways to do it, and some are fast and some are slow. And because of this, we have this issue where there's always this different ways. Some are easy to understand and easy to read, and others are fast but hard to figure out. And here in that case, because the matrix is still quite manageable, it holds only 12 rows, it doesn't matter what we use. For other stuff, it might be that we need to think about what to do fast, or we switch over to Python. And also because I've learned R before Tidyverse came out, I have a tendency to always fall back to these old-fashioned things. But the bigger issue of the exercise is that we've now here learned a bit on how to do some quality control and what we can now do next. If we then later want to figure out how to compare ourselves, we might now here make a break and say, let's cut around here and say all these cells here are reliably assigned to a sample, and for those cells we don't really know what sample it is from. So this was one thing. The other, this was one thing that we skipped over last time when we went through this code. You remember we just put these gene expressions here and put this just away into some count tags, and then instead we went back to our actual UMI counts. And the UMI counts, so what we've done so far now was a bit of metadata. We only learned about what sample it comes from, but not what's going on biologically in the cell. So let's instead go back to what we've done there. So you remember we had our count matrix, which again has the same columns for the same cells, but now the rows are no longer hashtags, but they are the actual genes. And you remember this matrix was really sparse. Now let's have a look at what we've done here. And the first thing is we looked at this histogram, and we noticed already last time that the minimum call sums, so here, for example, you have this issue here. I have written call sums count to get my column sums, and I also could have written apply counts, to, sum, which would have given me exactly the same thing, but you see, it takes longer. And that's exactly the issue, why we often use these special made things, because R is not clever enough to figure out, oh, I could branch over to the other thing. So I interrupt this here and go back to the call sums, which, as you can see, works in a reasonable speed. But I have to know that there is not only the generic way, but a special function for call sums, but it only exists for sums. It doesn't exist for maximum. So you then always spend your time Googling about for which things has somebody written a proper fast function for which it hasn't. For all these sparse matrices, there is this sparse matrix stats, which contain all these call sums and call means and call variances. And interestingly enough, it has been written by Konstantin, who once was, you know him, right? Who once was a master student in my group and is now up at EMBL. And he got so annoyed with waiting for this that he actually wrote the package that we now all use for this. So here, this is code written by Konstantin, which runs with call sums. At least I think it was him. So what we noticed last time is when we look at the minimum of a call sum, it's exactly 500. So my colleague who has run Cell Ranger has just decided to only store those cells which have at least 500 counts, and this thing was too low, because if you look at this histogram of a call sums, you see 500 is here. But even at 1,000, it isn't. Who remembers what this dip means? Why do we have one big pile here and a lot of things with really small counts here? Have I explained this? Who remembers where this comes from? Remember how in the 10x machine, we always combine in these droplets one cell with one bead. But we want to make sure that it hardly ever happens that there is less, that there's 2 or 3 cells in one droplet. So we dilute the cells and make sure that only very few cells come in. so that it never happens that two cells end up in the same droplet, and this means that most of the droplets will be empty. So we try to load the thing to make sure that we have, typically you put 20,000 cells in there, and then you can be sure that you get something like 10%, then you get something like 90% of the droplets are empty and 10% of the droplets have a cell. But the empty droplets, they still contain a bead, and hence they have a barcode, and we sequence these barcodes. And we sequence if you reach for these barcodes, because even if a droplet is empty, it still contains RNA, because during our preparation, a couple of cells break open and the RNA leaks out and just spreads everywhere. So we have to assume that in every droplet, there's the RNA of this actual cell and also some ambient RNA, which has just leaked into the droplet from other cells which have broken up earlier. And from this thing, we can conclude that the amount of ambient RNA is probably around a few hundred, but only once we have more than, say, 2,000 counts, we can be sure that there's actually a cell. So probably these are droplets which contain a cell, and these are droplets which only contain ambient RNA. We can conclude from this, unfortunately it's cut off, so we don't know where the maximum would be, but it seems that typically the amount of ambient RNA in a droplet is something between 100 and 1,000 counts. But only once we have more than 2,000 counts, we can be sure that this is actually a cell. Why 2,000? Let's mark here. It's always a bit hard to see on this log scale where 2,000 is. So I tell it here, I want a vertical line at the log 10 of 2,000. You see the log 10 of 2,000 is something like 3.3. And this is at the foot of this hill, and I can imagine if I extend this, it goes down. So I will cut here now. That's what I've done here. I say I only keep those columns in the count matrix where the column is above. Oh, I put my marker last time I did it, I put it at 1,500. Actually, I think 1,500 is a bit weak. Let's better say 2,000. So, and now I have reduced this thing, and instead of the 10,000 cells we have before, we now have only dim counts. We have only 19,000, and how many did we have before? I've forgotten, but probably a few more. So the next thing I now do is I normalize the counts, and here again, when we normalized here in this time, you see when we did here our little quality control for the hashtags, here we did the normalization. We divided the count for the which we are interested in by the total. And this time we do the normalization for all the counts, not only for the maximum count, and we divide here by the column sums. The column sums tell us the total counts for each cell. And we divide the counts. Who remembers why there's these two T's? Yeah. Exactly. R has this recycling rule, because this the first thing here is an n by m matrix, and this is an n vector. So the two arguments here have different number of things, and R has this rule that it just recycles the smaller of the two data structures. By recycling, I mean go back to the beginning and start again, while it goes through the bigger matrix row by row, but we want to go through it column by column, so we have to transpose it, i.e. swap rows and columns, and so that we really divide the columns or not the rows, and then we go back. Whenever I do this, I'm always worried that I get it wrong with the transition. So what I always then do is that afterwards I calculate the col sums again, counts, and as you can see, no, here are the col sums before, and now I calculate the col sums of my normalized counts and see they are all one, i.e. normalization was correct. Which, yeah, should be, but because I keep forgetting about whether it goes by row or by column, I always check. And then the next thing we do is we take the logarithm of this in order to get it on a homoscedastic scale. To see a bit why we need this logarithm, let me look at this and let me look at the histogram of the counts. So here you now see, sorry, that was wrong. If I take a histogram of all the counts, numeric. Okay, I will now not explain what this add x is, but just show you the histogram. And as you can see not much. I will explain the add x as just all the non-zero elements in this matrix. But even so, it looks they're all zero. I can take a logarithm of that to see something. Now we can see typically we count entries in this matrix. Most of them are around 1, 2, 3. A few are around 10. Hardly anything is ever around 100. If I do the same now with the normalized counts, then it's of course a little bit below. But you see, I had to take the logarithm here to get a nice histogram where this is nicely worked. But I want to see this in the actual, for the later calculation, I actually want to live in this normalized, logarithmized world. That's why we took the logarithm. And now I get everything logarithmized, and now I can look here at the counts without this. And we can now see here, these are now our logarithmic counts on a, not in a log 10 scale, but on a log E scale. And it has here, it's sort of spread in a reasonable number of values. So let's go quickly through the rest, which we've already done. So we calculated here the means and the variances, and I still wanted to explain to you what actually is happening here. But we do this next time, because now I want to go here into the PCA, want to go through this once. And what do we do first? Yes, we go to the end of this thing so that you see what comes next. So you remember the point of the PCA was to reduce the data that we have for the cells from this 30,000 dimensional vector to something with only 50 dimensions here. And why is my computer now so slow? The PCA, usually it only takes a few seconds, but sometimes it gets a bit longer. Which is good. I've talked now nearly 50 minutes anyway, so we might need a short break, which my computer takes by being slow. Actually, the server being slow, but this is now getting really slow. I interrupt it. Now we see an annoying habit of R. Many of these functions cannot properly be interrupted. So what I will do is just to restart R completely. See if R is alive again. Try this again. And now simply rerun the whole code while I sit back and wait a minute and relax a moment. In the meantime, what we can talk about is, I think last time we came up to the UMAP and looked at the UMAP. And then the next thing we usually want to do with the UMAP is to do a bit of, find clusters of cells which are close to each other. one thing you can already do while we wait for my computer is to install Sleepwalk, which is, I show you how this is written, because we will need this in a moment. Install packages Sleepwalk. This shouldn't take too long, but why do I? Okay, while we do this, I can look here at my new toy. Log counts, chain means, chain vars. Okay, now we are back at the PCA, which makes a bit. By the way, who knows how a PCA works and how it can be understood as a rotation? Okay. So when I use my waiting time to show you a first little demonstration of what a PCA does, and a great way to understand a PCA involves having a three-dimensional object, and imagine we want to project it into 2D. And so I hold up this bottle now. And to project it into 2D, what I can do is put it up here, and now you see the 2D projection of this bottle. And I can rotate this bottle, and depending how I rotate it, you either see a large amount of variance of all the points. So now you see the maximum structure, or if I rotate it like this, I see much less structure about this. So what the PCA does, it tries to find a rotation such that the projection retains maximum variance. So if you imagine each point on this bottle, this bottle is a point cloud, when you can calculate in the x and in the y direction the variance of this, the variance of the coordinates of these things. And if you do this in 3 dimensions, when you see, when each point here, you have the x, the y, and the z coordinates for all the points, which I have now drawn or have not drawn on this bottle. And you can imagine that there is a total amount of variance, and if I now don't only add up the variance of the x and the y coordinates, but not of the z, I get less variance. And if I now say I want to retain as much information as possible while going from 3 to 2 direction, it's of course better to omit this axis than to omit that axis, because this axis contains less information than that axis. And in this way, a PCA can be understood as finding the rotation such that the 2 axes which are then left have maximum variance. So we have to retain this axis and that axis and skip this axis, as you can see here when I move this thing. Now, I've recorded this, so probably whoever listens to this recording, I've been holding up a bottle, but sorry, you can't see what else has happened there. And in that case, we will later then think about it, and we can try this, I don't know, maybe next time, where we take this, where we imagine, of course, instead of going from 3D to 2D, we now went from these 30,000 dimensions to 50 dimensions, and the PCA has sorted our dimensions in an order that the first dimension contains maximum variance and the second dimension is a bit less. And here, in the output of this PCA function, where is it? Here, PCA dollar, we have here this the total variance, which is how much variance was in there beforehand. Strange that this is such a round number, actually. And here you have the variance, or actually the square root of the variance, the standard deviation along each column. So the first column so the first dimension contains 152 variance unit, the next 124, and so on. And you can already see with the first 10 or so variants I can explain a lot, and even the first two here explain a decent amount of the total variance, which means that if I plot only the first two things, PCA $x the first dimension and PCA $x the second dimension, I can already see a clear distinction of different cell population. Can say these are probably different types of cells. And of course, if I added, and of course, if I did this now in 3, 4, 5 dimensions, I see a bit more. While if I were to just take two random genes and plot them against each other, so for example, ln counts first one random gene and the other random gene, I wouldn't see anything useful at all. So the other thing, but still, ideally, it would be to not only look at the first two, but at the first 50 dimensions altogether. So what we try next now is that we assume that we take each cell and ask what is the distance of this cell to every other cell. And I just pick a random cell now. So see here, pick random cell 1, 3, 2, 5, and I pick this cell PCA $x, alt, this is the row, so this is my random cell, and I want to see how far this random cell is from all other cells. Distance of all other cells to cell I. How do I do this? If I have one random other cell, let's say the cell 1458, and this cell here, then I can just subtract them, square them, sum them up, square them, sum up the squares, and take the square root. That's the Euclidean distance. Who's familiar with Euclidean distance? Okay, great. So here we have a Euclidean distance between cell 1325 and cell 1458 is 49. I now want to do this for all cells. So what I do is I go through all the cells, 1, 2, and row. Actually, this could be a quick exercise for you. Make a vector which gives us the distance of our randomly picked cell to all other cells. So this vector then should, in its position 1458, it should have this number. It's good when I don't have to talk so much if I occasionally give you some programming. So this was to distance of cell I to random other cell J. And J is then 1458. Sends to all cell I to all other cells. So who has it? No, let be curious and see what different things you guys try. Everybody's still installing stuff. And we have to wait a moment anyway. About you. We have, of course, the problem here that we are all mixed together from different years and things, so I have no clue for how much programming ability you guys have. So let's look a bit. So what you would usually use if you do it in old-fashioned R is sapply. Who has used sapply before? Or who knows about sapply? Oh, now we have an issue. Nobody knows about sapply. We can also use the normal apply function. Or the map stuff. Okay, who knows map? That works as well. But Philip is too modern here. So, sapply. How does sapply work? Or you can also use map. It's more or less the same. So, in sapply, you take a vector, for example, C2, 5, 7, 9, 4.5, and a function, for example, square root. And what this does, it applies the function to each element of a vector. So, sapply vec, fun applies fun to each element of vec. So this is what we usually do. Now, in our case, we have here this function here. We can consider this here a function of j. So typically, the first, here you see I just used a function name. If the function is not named, can use an anonymous function. So for example, add 3. So we want to add to each element here the value 3. So I can make an anonymous function. I write function x, and then I say what it should do, namely x plus 3. And then I get this here. So far, makes sense? And often one uses index vectors. So I use, for example, sapply 1 to, let's take n call L counts. Let's use this here, or let's use n row, PCA $x. So what is this here? PCA $x is our PCA matrix, which gives us for each cell its 50 PCA coordinates. N row is the number of rows, so the number of cells, 19,000. And 1 to n row is the indices of all the cells. And now I can use this and index the cells, PCA $x, I, comma. This here does something with that here. For example, some G sum the PCA components for each cell. So, and now you see for each cell I've just added up these 50 numbers. It's not very useful, which is not what we wanted. What we now actually want to do, now use this to get a distance. So, who has an idea how I modify this function so that the calculation that it does is the one I use up here. Here we have a calculation of the distance from cell I to cell J, and now we want it here for each cell we figure out this thing. When you do this, don't get confused about our indices. I is this number here, and so and J should be the number which runs. So I give you a moment to try to sort of combine this row together with that row, so that you can put the two together and can see how to make one thing out of it. And that takes a bit of practice, of course, always apply and S-apply and so on, but it's quite essential to get this practice. And then, as I said, because once you've mastered that, you can more or less get R to do everything you wanted.